Section 7. Expert Opinions on the Current State of the Field. Given that variant classification guidelines rely heavily on expert opinion, it is fair to consider expert opinion when exploring the strengths and limitations of the guidelines. The expert opinions shared below are informed mainly by germline testing, but many of the comments also relate to somatic settings. Elaine, Lyon, and Madhuri R. Hegde were among the co-authors of both the 2015 ACMG-AMP and the 2007 ACMG guidelines for germline sequence variant classification. They remain active leaders in the global clinical genetics community and continue to develop protocols in their own clinical laboratories. For additional perspective on current practices, Christine M. Stanley, Iain D. Kerr, and Julie M. Eggington comment. Christine M. Stanley is a board-certified laboratory director, and Iain D. Kerr is a structural biologist. Christine Stanley and Iain Kerr are both leaders in high-throughput commercial variant classification, and Julie Eggington has extensive experience in quality checking and consulting on germline and somatic clinical variant classification pipelines, primarily in the high-throughput commercial laboratory space. The commentary provided by the authors is their own. Some authors may not agree with the comments of other authors, and the comments do not necessarily reflect the opinions of the author's employers or the organizations, societies, or committees of which they are a part. The authors also reserve the right to change their opinions. Section 7.1. The 2015 Germline Variant Classification Standards and Guidelines from the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. 7.1.1. Historical context for the creation of the guidelines. Madhuri, Hegde. A major driving force for creating the 2015 guidelines for variant classification was reductions in the cost of sequencing. Subsequent to this affordability, we began to encounter a rise in population data, necessitating rigor in how we approached variant classification. We really needed to look at all the variants in these population data because many of them would be benign. Elaine, Lyon. The variant classification landscape prior to the 2015 publication of the sequence variant guidelines truthfully did not change too much. Previously, we lacked a detailed standard operating procedure. We basically took into consideration different ideas and some differences in the strength of evidence and how to apply it occurred. One of the surprising aspects of composing the 2015 guidelines was realizing that across laboratories, we had orthogonally come to similar conclusions. Creating the 2015 guidelines was a process, and some concepts we could not address completely, but we could at least place those concepts in a framework where other members of the broader variant classification community could address them. Though some confusion persists on how to use the guidelines, I am happy with how they turned out as an important first step. In a way, the guidelines started the science of variant classification and interpretation. 7.1.2. Major successes of the guidelines. Madhuri, Hegde. The uniform acceptability of the guidelines was wonderful to see because the committee spent a lot of time creating them, including 2 years of debate and dedicated thoroughness. Additionally, I am excited that the community wants to debate the guidelines. It is critical for the progress of the field that people take pieces of the guidelines and debate them. I receive global feedback from colleagues both agreeing and disagreeing with aspects of the guidelines. I have seen that clinical decisions can drive the interpretation of a VUS, which can then engender its own debate, lack of endorsement from some for such decisions, and rebuttal, which is essential for progress. Christine Stanley. I most appreciated the premise of bringing initial structure and standardization to the classification of variants at a time when NGS was ramping up heavily, and therefore the number of variants for classification was exploding. Without proper guidance and established expectations in the field, we might have set genetic testing back years, because if labs were arriving at different conclusions, then the utility of genetic testing as a whole may have been seriously questioned. The straightforward definitions of the lines of evidence used in variant classification, coupled with good explanations and examples, served the industry well in the beginning to allow for adaptation, rather than an overburdensome and overcomplicated system, which may have delayed adoption. I preferred the 5-tier system rather than more complicated proposals, and the option to weight the lines of evidence up or down to prevent the under or overclassification of variants. Section 7.1.3 Implementation Challenges of the Guidelines. Elaine, Lyon. After the 2015 guidelines came out and were implemented, we were better able to see which components elicited more debate and, in some cases, more confusion. This indicated areas where we needed to provide improved clarity. Since the committee that created the 2015 guidelines was so experienced in variant classification, perhaps we did not anticipate the questions some of our less experienced colleagues had. Madhuri, Hegde. A criticism from the overall community that we sometimes receive is that the 2015 guidelines do not apply to one particular gene or another, even though the guidelines are intended to be more generic. Specific diseases, genes, variants, and heterogeneity need to be considered individually. I am uncertain if creating a guideline for every gene, which is happening right now, is going to be useful for clinical labs. Software and artificial intelligence will be necessary to support this, and so will the application of one skill set to variant interpretation. Potential future over-reliance on software concerns me. Moving forward, clinical lab directors need to play an important role, especially as in my experience on a CAP committee and in NIH proficiency testing. Most errors arrive from labs using automated software to classify variants. The software cannot do it all. Also, when evaluating evidence and publications, the whole paper needs to be taken into consideration. Piecemealing ought not have a role in these assessments, and I would argue that it can even be dangerous to segregate everything into a million pieces. Christine Stanley. Some of the challenges we faced with adoption was a lack of clarification of how to find the data for the recommended lines of evidence and how to utilize some of the lines of evidence. For example, co-occurrence evidence is very challenging. In practice, this led to using a subset of the available lines of evidence. Additionally, we needed a bit more guidance on using the scoring algorithm for cases when there was contradiction. For example, when there were 2 lines of evidence for benign, guidelines indicate the variant as benign, even when there is conflicting evidence. Also, there was some confusion in selecting lines of evidence of the same type, which amounts to double dipping and overclassification. We also were challenged with transcript selection. When reviewing variants, it was tempting to be overly cautious and select the longest transcript, or the one with the highest variant effect predictor score. This resulted in some cases of overscoring a variant as pathogenic, by using a transcript that was not one that was of biological significance. However, overall this was a great first draft, and there will always be a plan to update this first draft after the genetics community worked on it. 7.1.4 Application of the guidelines beyond the United States. Madhuri, Hegde. We presented the guidelines on variant classification internationally and have discovered that different countries take very different approaches to the guidelines. For example, in India, they think these guidelines are too stringent, perhaps because many clinicians practice medicine based on their skill set and are not reliant on software because it is not available. In this context, understanding the interpretation of pathogenicity can mean proceeding with treatment, as there is no mechanism to provide feedback through the guidelines. Furthermore, the practice of genetics in Europe is quite distinct from the practice of genetics in the United States. Other than 1 or 2 companies, whole genome sequencing has not been adopted. The debates that arise from this global perspective are critical. Sometimes in the United States, we tend to narrow our focus to how we practice medicine. We have an opportunity to evaluate these guidelines in new contexts when we are open to global feedback.